So I'm quickly going to describe what we're going to do out in the OESF this summer with our phase 2. So with the work that's going on with SEMR, with the forested wetlands project that they're starting up and stuff like this, and some stuff that's happening on the Oregon coast, looking at these small wetland features, we thought it was a good time to start up phase 2. So going out in the OESF, for our phase 2, we came up with some research questions here. With the first one, we're looking at stand age, probably using broad classifications of early, mid, and late Searle to evaluate that influence. As Jack just described, most of the wetland features are along the channel, but sometimes you get quite a few that are disconnected from the channel out in the riparian area. For number 3, that's just from our basic metrics that we'll get from our mapping procedures. For number 4, as Jack just explained, the stream temperature wetland relationship just needs more evaluation. So for 5, we're going to look at amphibians this time, possibly look at macroinvertebrates. So as you guys probably know, the big deal here is with the forest practice requirements for sensitive sites on type N streams. Okay, so listing here are some conditions that might influence a site's its sensitivities, things like aspect, forest age, gradient. Some recent work that's coming from the Oregon coast will add in geology to this, because geology is a major influence. But it's basically, as you probably know, they require a 50-foot buffer around these sensitive sites on type N streams. So in Oregon, actually, just an equipment exclusion zone is adequate in most cases. So it kind of begs the question, can you get away with less protection and be okay, given a specific site and its different factors that it's influencing it? So we're going to do this out in the OESF Basin 584 that we had talked about earlier, the thermally sensitive one. Easy access, it's only 20 minutes from Forks, and the road system up there is great. We can get to most points within the basin. It's all managed by Washington DNR. We did a screen last year, excuse me, in the summer, looked at 25 basins, visited 15 of these, went out in mid-September when usually the stream channels are at their lowest. If you go out there in the spring, everything is wet. So Basin 584 rose to the top because it had exactly what we were looking for in terms of things that we can stratify in terms of aspect, forest age and gradient, and length of surface flow. So we chose that. What we found with the other basins, the other 15 out there, is basically these wetlands were hard to find, which was kind of surprising to us because in our phase 1, they're pretty common out there. The other thing we found with the other basins in terms of not choosing those is we're looking for headwater perennial streams. And so a lot of those didn't have those types of streams flowing through, different age classes and aspects and that sort of thing. So anyway, 584, it works with us. So what we're going to do out there is the same procedure as we did in phase 1, looking at surface hydrology, soil pits, and wetland indicator plants. But we're also going to put some temperature loggers out there as well in the lower portion of the streams. But before we do any mapping out there, we're going to survey for amphibians. So what this is a 50 meter reach along the channel, 5 perpendicular 2 meter wide transects. Each one of the transects is broken into 3 zones, looking at the bankful width going up each of the stream banks and out into the riparian area about 20 meters. The surveyors will be turning over rocks, logs, wood, stuff like this, kind of a light touch technique. And be taking weights, lengths on the amphibians. And anyway, these constrained areas are just like this, pretty standard fare for amphibian surveys. We're adapting some methods here that the P&W station used in the late 90s with the riparian study that was done all around the peninsula. So from that previous work, we pretty much know what we're going to find out there. You know, some species like the Cope's giant, pretty much fully aquatic. The torrent salamanders and the frogs, they spend a great deal of their life cycles in the water. Some are fully terrestrial like the Encetina. But most of these species kind of hang pretty close to water sources out there, especially during the summer when we're going to be doing our surveys. So just a tidbit from that previous work, we found that these 3 species in particular, really highly associated with water out there. Just for reference, the Van Dykes and the Western Redback, they're what we call plethodon salamanders. They're lungless. They breathe through their skin, so they need high moisture levels to be able to do that. Another aspect we want to look at is environmental DNA. As you guys probably know, it's becoming a pretty common survey method for population surveys of aquatic species anyway. And just in a nutshell, the DNA that's contained in feces, urine, skin cells, and this sort of thing that's in the environment can be collected in a filtered water sample, sent to a genomics lab where they can extract the DNA and sequence it and tell you what species are present. Another thing, our interest in doing this is basically because we want to compare what the DNA lab results tell us, and you usually get back a lab report that says DNA copies by species. So we want to compare that to what we're getting from our surveyed amphibian abundances. The DNA technique, it's very quick, it's very inexpensive. We just want to get an idea of its feasibility for doing amphibian type survey work on type N streams. And that's kind of where we're going with this. So.